Well, hello everybody, and welcome to today's QAAFI seminar, which will be given by Edward Narayan from the School of Agriculture and Food Sciences. I'm Alan Tilbrook, and I'll be hosting today's seminar. Before we commence, I would like to acknowledge the traditional owners and their custodianship of the lands upon which we meet today, and pay respects to their ancestors and to their descendants. And if we have anyone who is listening to this seminar today who represents Australian Aboriginal or Torres Strait Islanders, we welcome you and we hope that you enjoy the seminar. Edward's seminar is going to go for about 40 minutes or so today. When you think of a question, and we hope you have plenty, please type them directly into the Q&A part of the webinar, not into the actual the chat function. And we'll address these questions at the end of Edward's talk. So it gives me a great pleasure to introduce Edward, who is a senior lecturer in the School of Agriculture and Food Sciences here at the University of Queensland. And Edward has got a pretty interesting track record. He completed his PhD at the University of South Pacific, where he pioneered non-invasive reproductive and stress measures of endocrinology in amphibians. And he's gone on and got a pretty extensive track record in postdoctoral research in 4 countries: India, New Zealand, Canada, and of course, Australia. And it gives me great pleasure to introduce you today, Edward. The topic of your talk is tracking stress remotely via wool cortisol and smart tags. Thanks, Edward.
Thank you, Alan, and thank you everyone for attending today's talk. Today I will be sharing my research as an animal biologist and a stress physiologist. The presentation itself will cover the journey that I have taken in the field, in the dynamic fields of animal welfare and productivity, and use sheep as an example. But before I continue, I'd like to share some snapshots of the various research experiences that I have had in Australia, Pacific, and Asia. Been lucky to work with many dedicated senior researchers, industry personnel, and HDR students. I'm hoping that the journey that I took will inspire young scholars here in Australia and internationally to take up opportunities as they arrive and make the most of postdoctoral opportunities before getting or landing a permanent faculty position. I think it's really useful. So we continue to face challenges for our very own existence due to human-induced impacts on the environment. Agriculture has a very pivotal role to play in the sustainability goals, and it continually gets questions for its methods of animals and plant production and the contributions to the environment, both on land and water and the oceans. So the context in which we are now at could drastically change over the next decade and years to come. And agriculture has a very pivotal role to play in all this. And hopefully we'll see how this all fares out. Here in Australia, we've had unprecedented environmental context even before COVID hit. As we know, we face a very severe and prolonged drought, and that leads to really downstream effects on soil health and pasture growth, as well as really damaging effects on the animals' health and well-being. And the context continues to change as we move into the spring and summer months. Now we are seeing heavy rains here in Brisbane, and which may be a good thing in the interim, but it could have drastic effects. So the outlook is a bit grim due to water shortage and extreme temperature forecast and drought in eastern and southern parts of Australia. The rainfall levels for the season were quite low, to historical lowest levels, up to 10%, which is really something to think about. It's not just here in Australia, but globally we are facing unprecedented times with regards to food security and being able to nourish our kind. So according to the World UN Food and Agriculture Organization, maize and rice and wheat together, they make up 50%, up to 51% of the world's calorie intake. And billions of people rely on this. And just imagine what would a disruption, a major disruption to the production have on the health and well-being of people around the world. So we are not just thinking about preparing for the now, but into the future, we are also trying to think about innovative ways to secure food security. And a lot of innovation is happening around the world, as you can see in some of the slides. The industry is projecting that the number of progeny is going down. So we've had reductions in lamb slaughter, lamb production, lamb export, sheep slaughter, and not only in the meat sector, but also in cotton and wool. We've had a bit of a reduction due to competition, but also due to the unpredictable market that we are sitting in right now, which you must have heard recently as well. But one of the other big factors that's actually playing a significant role in all this are the consumers. So the consumers are pretty much driving the change with regards to the supply chain and the product itself. So it's a very interesting time to be in. We can see that 10 years ago, there was a lot of interest in regards to the nutrition of product, in this case, meat product. But now we can see we can have a plus sign and add animal welfare into the mix as well. And here in Australia, we are leading the way with regards to animal well-being. And I'll hopefully share some examples through the next few slides and discussion. So what is animal welfare? Basically, animal welfare is about the state or the well-being of an animal. So it's basically the basic needs that are being met and suffering is minimized. But we also need to look at the other side of the coin, which is on animal rights. So it's a belief-based system that animals have the same rights as to life and liberty as what is afforded to mankind. There is a suite of issues in animal production, which we should discuss. And some of the things, like why should we raise and kill animals? Should we eat them? Should we harvest non-domesticated animals here in Australia, with kangaroos and all sorts of emerging industries? There's a few discussions around that as well. So should we also kill sentient animals? So what is sentient all about? Hopefully we'll discuss that in the next few slides. So animal welfare science actually hopes to provide an objective assessment to these questions, but obviously it cannot solve all of the ethical problems. The context is all around us, not just with livestock, but also with wildlife. And animal welfare has become an important part. It has always been an important part, but it's actually, we are being asked more questions with regards to animal health and well-being now more than ever. But it's the people's moral responsibility to ensure that the welfare has been cared for. And a lot of work has already been done in this area. For example, eliminating pain and injury. And you can see that the cascading positive effects of an intervention on the farm to manage stress can have cascading effects on the reduction in carcass damage, wastage, reducing bruising and injury, decreasing mortality. So the quality of the product eventually improves, which is important. And you also end up with better conditions. So it's not just about the animals' welfare, but the people who actually work with animals. That's, I think, even more important for the sector right now. So it's a continuum of contextual factors, emotions, subjective and objective views, but pretty much a community sense is also existing here, which is going to be vital with regards to carrying agriculture forward. When we look at the animal welfare paradigm, we've all heard about the 5 freedoms of animal well-being. Now what we hear about is the combination of mental state. So we want to tap into the emotion of animals to understand how they are feeling, which can be a bit challenging because being prey species, they often are good at hiding their behaviors. So what we are trying to do here with the research that I will share is hopefully some of the biomarkers that we can use to assess stress in particular that may be used to index acute and long-term stress in animals. So it's all about the mental state, and a lot of research has been done and is going on with regards to the limbic system, which is a very complex network of nerves and a nervous system that connects the brain with your behavior. So you can actually tap and do these high-end methods, but we are also trying to come up with some minimally invasive methods to actually capture that emotion of the animal. And as you know, that animals are highly social, highly connected. They recognize their carers, and they have a very soft feel for the person looking after them, and vice versa. So it's really important to understand that connectivity. There are some good examples out there already. So you can see that even stroking by a human of lambs, you can see the white bars and the black bars, which depicts the heart rate on the y-axis in beats per minute of lambs either being immobile or being stroked by their carers. You can see that the heart rate actually goes down, and heart rate could be a very straightforward indicator of positive response to caring of livestock. But like Temple Grandin says, Professor Temple Grandin, I've been lucky to have some interactions with her a few years ago, and she says that we need to calm down in sync with the cattle. So it's really important to be able to understand the animal that's in front of you with the help of its senses, other than the linguistic senses that we use, but they also vocalize and they move their body parts in different manner. And so some of this technology has been validated, and you can look at things like ear and eye and nose communication of emotion. Stroking of cattle, like I said, can reduce fear and increase calmness. And you can also look at the visible white of the eyes, to index whether the cattle is relaxed or not. What I really believe in is this whole farm to the plate approach, and it is also a core aspect of the Australian beef sustainability framework, of which I was lucky to be a small part of recently. They have been having a lot of discussions around this. So animal welfare, as you can see, is one of the main pillars in combination with economics, environment, and the community. So if you look at the 6 main themes: animal husbandry, the antimicrobial stewardship, even the health and safety of people, balance and tree cover, they work hand in hand with regards to the end product. So we need to be very careful with regards to how we assess these things and have a very good on-farm balance sheet of sustainability, I guess. So going straight into now in a bit on the stress research that I do. So, stress can be a variety of factors that can influence animal growth, things like microorganisms, parasites, housing and transportation, even the day length, temperature and toxins. And what you actually tend to see is that the nutritional intake of animals can be reduced due to undue stress. And that could be a direct signs of stress operating in that particular system. We can look at things like physical stressors, psychological stresses. So psychological stresses that are actually animal to animal. Maybe you have a bigger animal with a smaller animal. As you know, animals are highly temperamental, and you can have a bit of a hierarchy going around with regards to the social nexus of the animal. So it's really important to be able to know these things, and it's going to be useful as well. So we can distinguish them as physical, environmental, metabolic, or psychological, and they can have various effects on the animal. And there are similarities in the stress responses across the species. For example, birds will actually stop socializing, so they stop calling. We're doing research on koalas as well, and you can see that they are actually stopping to call when they are overly stressed out in the wild, or frogs stop singing. But what does a sheep do? Let's look at that. So stress basically, as I said, could be anything like temperature, poor ventilation, overcrowding. So these are things right in front of you. If you're standing in a shed or a paddock, just think of what are some of these physical and psychological stressors that are operating right now in the system. And these are some of things, even like human exposure and pests, as we know, feral animals can be a huge burden on our animals. And basically what it generates is a stress response. As you can see here, the hypothalamus and the pituitary gland connected to the adrenal gland. So it's the HPA axis, and glucocorticoids are a suite of stress endocrine biomarkers that's actually secreted, and it actually helps on the acute scale to mobilize energy that can help the animal to move away from the stress. So an acute stress response for a wild animal is not necessarily a bad thing. For example, if a frog needs to escape a snake, but think about what it could have in regards to a sheep or a cattle. Maybe a cattle is destined for the slaughterhouse. What that acute stress, as in the minute changes in the stress response, could have on the animal. In this case, we are interested in the animal's productivity, but also its wellness. So this whole stress endocrine system is a network of neuroendocrine nervous system interconnections that helps the animal to cope with acute stress. So we've looked at some of these acute stresses in the previous slides, and it basically generates an alarm response. So you can see Sean the sheep is very happy over there in that slide, and it generates an alarm response. And basically you end up with full adaptation if the stressor is benign. So what could be an example of this? Now, we've had a lot of discussions around shearing as an acute stress. Yes, it is an acute stress. It's not a chronic stress. And this has been beautifully demonstrated that it actually leads to an acute increase in plasma cortisol, which basically activates this, is a sign of the activation of the stress response. And then it leads to acute hypothermia. But then there is no further effects on the health and well-being of the animal. So the research on shearing and stress that I will discuss later on is not looking at acute stress, but the long-term picture of how we are now able to track stress in wool samples. So we'll talk about that as well. Another good example of acute stress, which I thought could be valuable for us in the crowd, is this whole story around the glycogen bucket and stress. So, we know the input is nutrition, and muscle glycogen is really important with regards to the output of the meat quality, one of the suite of indicators that is being used to index meat quality by MLA. And we can see that the pH is a very fine interaction between glycogen and pH. And if you have stresses in the system, it can actually ruin the product quality. So we've been lucky to be involved in some of this research over the years, and we can see that the ultimate pH, for example, is an index of one of the indicators of meat quality, if you can call it, and then you can see fecal cortisol metabolites. There seem to be some sort of a correlation between pH and meat quality. So you could actually use that. And in this case, we used second crossed lambs to demonstrate a correlation between post-slaughter fecal cortisol and the meat quality, in this case, PHU, which is an indicator of dark cutting, which here you can see there's a positive correlation that indicates that if there's more input on the farm on stress, it could potentially have a downstream impact on the meat quality itself. So it's really something I'm fascinated with. But when you look at chronic stress, what are some of the examples of chronic stress in livestock? In the wildlife world, we have really good examples. For example, the work we've done with bile farming in bears in Vietnam. They are classical case of chronic stress where you see huge emaciation of animals, really unable to integrate with the, within the sanctuaries. And it's really something here in the contrary to acute stress, you can see the alarm phase is activated, but then the adaptation is not full enough. So it's actually incomplete adaptation. Shown the shape is sad, unfortunately, and you can see it's a loss of fitness, losing to eventual exhaustion. A really classical example of this is the work done at CSIRO a few years back by a distinguished endocrinologist, Ian Kozlist, and he demonstrated in sheep with fly strike that exposure to fly strike itself leads to elevation in cortisol. But not only that, if you look at the boxes on your right, top right, you can see the second row, the top right, plasma cortisol. If you can see, there's like a hill of cortisol elevated in the fly struck sheep. You can see that not only that, there is elevation in secondary immune markers such as IL-6 and serum amyloid A and hepatoglobin. And you can see that they are all elevated over the duration of exposure to fly strike in this shape. But what is the consequence at the end that makes me think that this is chronic stress? Obviously, the physical signs and the metabolic consequences are there, but downstream to productivity, you can see that there is a reduction in staple strength of the wool itself. And so there was a significant reduction in the wool staple strength of the sheep that were struck. So I think these examples provide a good examples of distinguishing between acute and chronic stress, which I think will be really important for us researchers to be able to deliver something useful for the industry down the track, because both acute and chronic stress are actually working in a nexus of the stress response and that whole animal well-being paradigm. Essentially, it's not just stress. It is one of the suite of biomarkers, but that's beyond of my presentation today. So let's look at measuring stress. So how do we measure glucocorticoids? So basically, glucocorticoid is the stress hormone, and we can measure that in blood samples, which is a very important feature of veterinary diagnostic testing, and it's really important. But it essentially provides an erratic snapshot. It's current. It's a very specific steroid molecule. It's sort of, you get the kit from the shelf and you'd measure the total cortisol. Now, remember, in the blood itself, cortisol is bound to the bounding globulins. Now, in cases of the bear that I mentioned before, their cortisol is actually freely circulating in the blood over a long duration of time. So with chronic stress, you could really bugger up that whole binding globulin effect as well. And there is evidence in biomedical literature as well that chronic stress can actually ruin the binding globulins and affect the stability of cortisol in the blood, because you don't want to circulate the cortisol freely all the time. Otherwise, it could be self-damaging. So it can be a bit limiting if you want to track these things more regularly in livestock, in a farm context, or in a feedlot setting. You have a mob of cattle that has just arrived, and you want to break them in and sort of monitor them. I guess using these minimally invasive samples could provide a more interesting picture, because it gives you a general summary. It's retrospective. We talk about metabolites, not just the original steroid, because the stress response is generated, the metabolism happens, and then it tends to be collected in the excreta. And it also provides for continuous sampling, and it possibly is more ethical friendly, which might be more favorable for animal ethics applications down the track. But anyway, when we can see over here that the cortisol in the blood, as I said before, is binded to the globulins, but then it goes through a suite of events. You can see metabolism happens in the liver, it's conjugated, then ends up in urine. But then if it's actually continuing further through the GI tract, it becomes metabolized by the bacteria as well, and then eventually ends up in feces. So when you're looking at minimally invasive hormone monitoring, you need to be very much aware of what you are looking at and what the theory behind this is. It's really important before you go and collect a kit from the shelf and just measure and get a number and run with it. It can be a bit dangerous. So we've been working with non-invasive or minimally invasive hormone monitoring for a little while now. And this is Josh in our lab here. And we've validated one of the polyclonal antibodies. So you can see it's called the R4866 antibody, which is polyclonal, as in it will be able to sequester a range of metabolites in the excreta with regards to the cortisol being the main ring in the main central moiety, but the side chains are all different due to this deconjugation and bacterial metabolism. But if you have access to a system like this, then what essentially you can do is you can have a better power in terms of your assessment itself. So there is a suite of laboratory tests that you use to validate the method. Here you can see a parallelism of fecal extracts of sheep feces and cortisol standard, which is the polyclonal cortisol antibody. And it basically shows a parallel like a railway line. If it crossed each other, then it would not walk, essentially. One of the other things we've looked at is to see the natural weathering of cortisol metabolites in the feces itself. So here we can demonstrate that the skates are stable for a few days. This was done in a bit of a winterish month in Wagga at Charles Sturt Uni when I was over there. And basically we can see that, if you look at 0, that's day 0 of exposure, day 1 on the x-axis, and then you can see some outliers, and then there is an X-strict on day 5. So the good thing about sheep feces is that it tends to be quite stable on the farm. So you can actually pick up a handful of scat, bag it, and send it to the lab. And the other interesting result from this research was that the intra-sample variation is quite low. So a single subsample can be adequate for reliable detection of fecal cortisol metabolites. And if necessary, a subsample can be taken from any location from the fecal mass. And it's also important how you take the feces, whether it's from the ground or directly using a standard operating procedure from the rectum. So these few slides will just showcase to you the validation in a way, but also the application of this methodology, which I think is really important, because to validate it pharmacologically, you'd essentially want to subject the sheep to an ACTH stimulation test or a dexamethasone suppression test. In our previous research in Wagga, we were not able to do that, but anyway, we went ahead with the laboratory validation, which I demonstrated. And also here, being lucky to be a part of this big research done by researchers at CSU, who looked at perennial ryegrass toxicosis, which is a disease caused by a mix of toxins, including Loretrum B, produced by a fungal endophyte. And it's basically a tremogenic neurotoxin that is thought to have effects, neurological effects, by blocking the calcium ion activated channels in the sheep. And in this case, you can see it will lead to sloughing of the skin. So it could have really high and huge animal well-being impacts. The sheep is unable to feed, unable to see, and it's really self-damaging on the animal. So with this ryegrass toxicosis being a huge problem and it leads to economic losses in sheep and cattle, we, as part of this research, just a graph to demonstrate how in feces we were able to detect that acute stress response in a treatment group, which was actually exposed to the ryegrass, and in control group with just the ryegrass itself. So cortisol as a systemic marker of stress in lambs could be useful in the field to assess as an early warning tool. Could it be used as an early warning tool for farmers who could then institute altered management practices or therapeutic interventions prior to manifestation of this, of this condition? So basically what I'm trying to say is that using this tool, we can actually early index the stress status of animals. And I think that would be interesting if you are able to have an on-farm, on-gate approach where we can sort animals with regards to their stress levels. I think that would be really cool if we could do that. So now I guess it goes to the big story, the elephant in the room in a way that has been on the news last week as well. Pregnant sheep shown early in pregnancy, whether it improves productivity. So this was a research I ventured on a few years ago with colleagues in New South Wales as well. And it was a very interesting research because I'm really interested in understanding how the connectivity between the mother and the fetus operates under stress. As we know, in pregnancy, there are two bodies inside one another and people live under one skin. So we can see a suite of factors like grazing, maternal stress, body condition, lamb quality and survival. So over roughly 152 days of pregnancy, the production output is a combination of genetics, the environment, and nutrition. So nutrition in sense of being managed, looking at the management and the well-being of the animal. So one of the earliest research we sort of did was to look at in the AI world, artificial insemination world, in donor ewes, research done at Roseville Park, one of the biggest producers of superfine Merinos here in the country, and highly dedicated producers who are really interested in research, which is fantastic. In this study, we actually looked at in donor ewes, does the embryo quality actually reduce drastically at a certain temperature? And interestingly, we saw that under 39.5 degrees Celsius, so these were rectal temperatures, the embryo quality, so the number of percentage transferable embryos was 89%. But as soon as the ewes were actually over 39.5 degrees Celsius, there was a drastic reduction in the observation of viable embryos. So the embryos became highly non-viable. So what's the reason behind this? What could be some of the factors that interplays with regards to the loss of embryo quality? As we know, this is termed as reproductive wastage. So heat stress is postulated to affect the folliculogenesis, as it has been shown to suppress follicular development and reduce estradiol B concentration in the follicular fluid and prevent the dominant ovarian follicle from exerting dominance. This has been shown in cattle. So oocyte competence is believed to be compromised by maternal stress by impairing the folliculogenesis, which can result in ovulation of aged follicles that contain the oocytes with reduced competence, and hence they are unable to develop properly after fertilization. So it has been demonstrated in cattle, and further research in sheep, I think, is also ongoing. So do cool ewes perform better? That is the question that we are trying to explore as well at the moment. But we also saw that the percentage transferable embryos in the donor ewes, now we are looking at one side of the coin right now, just looking at the donor ewes. I'd be interested in tracking the whole development and then looking at the receipt ewes as well, and then looking at the bigger picture to see what is happening. And that is something that I'm really interested in. You can see here that there was a negative correlation between percentage transferable embryo and fecal cortisol levels in those donor ewes. And this research also was discussed in the land. Interestingly, the same graphs were used for this discussion, which was interesting. So now I guess I'd like to talk a bit about wool and the connectivity between wool and wool quality and wool cortisol. I have sort of been lucky to be a part of this research recently, and it seems to me that wool can be a very interesting biomarker to track long-term stress of the sheep. In this case, we are tracking the ewes, the female sheep, as opposed to the rams, but I'd be interested in the rams as well at some stage. But here, what you can see is an image of wool from the skin with 9 months of growth. So each line arbitrarily shows 9 lines with 9 months. A paper that we've sort of just we've been trying to get it published. Hopefully it will be published soon, but it's an interesting one. Again, in wool, we are seeing that if you look at the two graphs, there is a negative correlation between wool cortisol on the y-axis and wool macrons when the ewes are actually pre-parturition, so before lambing. That means they were starting to become stressed, and wool quality was sort of starting to be diminished, which is known that during reproduction, the investment into wool quality does go down. But here we are demonstrating that using wool, which has, to my knowledge, not been done before. But we can see here as well, once the lambs are dropped on the ground, then the relationship overturns itself, and you can see it's flipping over, even though the p-value is not significant. But it's interesting, the tracking of stress in the ewes, and I would be interested in looking at this from a perspective of feral pests, because with the lamb loss, the lambs can be taken away by a bird, or it could be a fox or something. Then we can actually see how the stress is tracking in the mothers, and how that could be used as a tool to track that as well. So it's really important to promote your research for the young people in the audience. We've been working with the media team at UQ to promote some of this work. And basically, what we tried to do in this research was to track stress and the activity of the ewes that were either shown all once or shown early in pregnancy. So we had two groups of Merino ewes, 48 of them all up. You can see the picture, and if you follow the purple arrows going down, one group, 24 of them were not shown early, and then the other group was shown early in pregnancy with preg scanning. And then we tracked them and then monitored their reproductive output through time. So what did we do to monitor their behavior was we used this tagging system that has been developed through DigiBile, is being used by producers all over in Australia quite widely right now, which is fantastic. But we used a very prototype state of that when our research was kicked off. And you can see these little dots are actual lambs on the ground, and you can have the same image on the producer's mobile phone, which is fantastic. So it actually depicts grazing behavior, standing or walking behavior of the sheep, which is fantastic. But in the wool itself, that's what my area is. And so I wanted to see if the grazing behavior of removing the wool early in pregnancy from a mob of ewes, does that influence her stress levels? Before I go into the box here to look at the results, on the image on your right is actually research done in cattle, and it was also done in pigs, a paper coming out actually this year. Is it getting into hair? So you can see here an image of cortisol with the red shaded color, and you can see the incorporation of cortisol, in the hair itself. And you can see that's a very good evidence that it is incorporated into the hair via the blood vessel. So that's a tick in the box, no doubt about that. Now looking at the results, you can see that pre-joining there is no significant differences. To some reason, the levels are quite low. To tell you, we used the top knot samples from the crown for these measurements. That can be discussed later on, but we can see later on that the late stage gestation of the twice shown ewes, their wool cortisol levels on the y-axis in nanograms per gram is actually significantly lower late in gestation. Not only that, we can see here that the body condition of the ewes that were shown twice, as in they were shown early in pregnancy, was much better than the condition score of the ewes that were shown once. So that was the second result. And the third result was that using the DigiBale ear tags, we were able to track. Now, this is the data I've showed, but hats off to my students and my team members. Like, it takes you night and day to be able to sequester the data from the model to be able to use it for research purposes. For a producer-centered application, that's not a problem, but for research, it's a different ball game altogether. And it took us a lot of heartache and day and night, blood and sweat to be able to get to here. We are still working on this. And essentially what you can see here is that the pie charts depict an increase in up to 10% grazing, the purple part of the pie chart. You can see that the grazing activity has increased by up to from 34 to 45. So that's telling me that the ewes are eating more once her wool is removed. Just imagine being you with the fetus in your tummy and with the full fleece. You can add 2 and 2 and understand that. But from a metabolic point of view, we can also see that the ewes on the graph on your right with the shearing frequency 2, the green line is actually tracking more. It's so beautiful, the technology. You can actually see, like, late at midnight, 0, and then 2 a.m. in the morning, they are hugged together. It was very cold, wintry morning. And then when the sun goes up, it's going up and starting to behave and graze. So it's really beautiful. I think the technology itself it seems to work, which is fantastic. But here you can see the green and purple lines are doing their own thing. They're tracking together. The red line is actually the ambient temperature. It wasn't massively hot that day, going up to 29 degrees maximum. But it's really interesting. We can now sort of better track the behavior of the ewes using remote sensing technology, which is fantastic. But from a production quality point of view, that is the main result of this whole research. And I think it needs to be understood very carefully. What you can see here is that the lambs at foot, we quantified the microns and other suite of wool indicators of these lambs. And what you can see here is that the wool macrons were becoming finer. Okay, so they became up to 5.0 of a 5-fold finer. But also the comfort factor has improved. You can see on the third last column, the ewes that were shown twice, their lambs have a comfort factor of 99.81 as opposed to 99.41. And the average spin fineness was also improved. But the other interesting fact is I was in a crossroad because my p-value was greater than 0.05. So this has been giving me a bit of a sleepless nights lately because the data is having a very interesting effect from a production perspective, but the biological significance seems to be quite important, but the statistical significance is something that was not there. And it's something that I have been trying to seek feedback from my mentors to understand how we can report this data and make it of use. One of the other things we are venturing into is looking at some of the epigenetic influence of management of livestock, and in this case, the ewes, using a collaboration with the Australian Genome Research Facility. We tracked the ewes and the lambs pre-lambing. And interestingly, we could not find any statistical significance of shearing on the epigenetic influence on the first generation. But I was still happy that we were able to quantify molecular marks in the mothers and the lambs as well, which gives me a bit of a baseline to track stress through time and better understand this with regards to other management factors or environmental factors, which will be interesting. So this work is going on. We are trying to track this in in the second generation, because that could be, but it could potentially be another presentation at another platform. In summary, basically what we saw was that the Digibel Smart Tech data showed us that the twice shown ewes spend 46% of their time grazing, as opposed to the once shown ewes that spend 35% of their time grazing. That extra feeding time actually converted into higher body condition score. And I have cross-checked this data with some leading wool producers as well. So the body condition scores of ewes shown once decreased continuously throughout pregnancy, while the twice shown ewes body condition score increased between pre-joining and mid-pregnancy, followed by a slight decrease between mid and late pregnancy. The higher body condition score of a twice shown ewe would also help to boost lamb survival rate. We did not see any significant difference between survival rate because our trial was very small. But because the mom is eating more, she is apparently happy. That was the twist, the hook that our media personnel said we should use, that they're happier. There is a greater plane of nutrition. So they are sitting on a better nutritional plane coming through, and the lamb is likely to benefit with the higher birth weight and better milk supply. So I would be really interested in quantifying milk indices. You know, there's various biomarkers in the milk itself. So I think this research has got grounds to move it to the next level. The assessment of the lambs showed that the lambs from the mom shown twice had finer wool by 0.8 of a micron, and had average higher comfort score and spin fineness, which is a difference of roughly approximately 0.9 of a micron. So the research is showing that shearing twice a year has a potential to reap immense benefits for wool growers in the right circumstances. So obviously, you know, tradition is meeting science, but the tradition is very important above anything else. If you shear every 6 months, let's say, for example, you could get 65 or 70 mm, but if you, that gives you roughly 140 mm per year. But in this context, you are actually getting a bit more. So I think that is really important. So when you look at the price of the wool, now you could go into the price, which is really important as well, the cost and price. But 16.9 macron wool is worth 1,846 cents of a kilogram. But in this case, at 16.3 macrons with a 0.5 of a fineness, it jumps to 2,030 cents. So the visual differences in our case was not statistically significant because of the small sample size. We are proud in a way that we do seem to match up with the views of astute wool producers. And it's really something that could be trialed as a phase 2 to replicate this with a larger mob, or even look at some of the other indices in the use, like milk quality and things like that, that goes to the lambs. And it also heads off to my former master's student, Greg Sawyer, who played a big role in this whole research. He's a wool agent and a wool and sheep researcher with over 30 years of experience. And I think this connectivity between a researcher and a person with industry connection is just godsend. It takes you even further with your research. The stress lab here at the Gatton campus is hoping to attract more students and get more research done in the next few years. We are also leading an animal welfare special issue, which now has over 82 authors around the world. So keep an eye out on that for the astute researchers out there. And you can follow my research on ResearchGate. And big thank you to my mentors, students, funding providers, network and collaborators, and Ellen for all his support and help during this journey as well. Thanks for listening, and any questions, please.
Thanks very much, Edward. Appreciate that. We've got time for just a couple of questions, and we've got two actually from Orlando, Edward. And The first question is, what sort of collar did you use, a GPS collar or a GPS plus accelerometer collar? And the second question, I'll give them both to you, is how often did you get the information from the collar? Please, Edward.
Thanks, Orlando. That's a very important question. We were getting data every 9th of a second. So that was a lot of data. This was a GPS combined with the accelerometer.
Thanks, Edward. Peter James has asked a question. Wool and hair are different fibres. Edward, did you measure cortisol in both types?
We haven't looked at hair specifically at the moment, Peter, but it's something that we'd like to do. Our team was confident that we were working with wool, but it's something that I'd like to pursue as well. Thanks.
Thanks, Edward. I have a question here from Robin Warner. She said, great presentation, thanks, Edward. Interested that Merino sheep had reduced stress and increased productivity if shorn early. Our research has shown Merinos have very different stress and metabolic response to second cross. Wonder if you could speculate on the difference for second cross ewes.
Yep, it's some. We were using Merino ewes. I believe they were super fine Merino ewes. We had, I think, up to 6 rams in the trial with very unique genetics, which some of them trace back to, I believe, 100 years or so. We haven't looked at the second cross at this stage, but it's something that I'm really fascinated with. And you'd see me doing more research in sheep and wool over the next few years to understand. There's a lot of questions to be answered, but the good thing is that wool seems to be a stable platform to deliver some of the biomarker technology and answer applied questions. So thanks, Rob, for your lovely question.
Thanks, Edward. Tim Marnie's actually asked a question about the HPA axis and wants to know when it becomes active in lambs, Edward, and would cortisol from the ewe be expected to act in the lamb?
Thanks, Tim. It's a very important question, I suppose, because the connectivity between the lambs and ewes, as I understand it, it starts to develop very early in conceptance. And I haven't read this in the lambs, but in the human fetus, the adrenal glands become activated in 14 to 16 weeks, I believe. Correct me if I'm wrong, sorry. It is possible that the maternal influence, and we've seen that like cases of, and that's why sheep is used as a model for things like intrauterine growth restriction and the brain partitioning of resources in early development. This is possibly a question that we should explore here in a more controlled setting, in a more discovery type research. But definitely I'd be keen to understand the connectivity between the ewes and lambs from a neuroendocrine perspective. I'm sure it is there.
Thanks, Edward. And perhaps the last question for today, given time, is from Cameron. Cameron, a very nice presentation. How many time points did you measure cortisol in a season?
Thanks, Cameron. I appreciate your feedback. We measured the cortisol 3 times. So pre-joining, early pregnancy, and mid-pregnancy. As you'd know, that the ewes would be scanned twice. So we took the opportunity on the farm to track and collect the wool samples. So it was looking at late or summer and entering into a bit into winter.
Thanks, Edward. I think we probably, in the interest of time, will close it out there, Edward. But I would like, on behalf of everybody that's listened today and from QAAFI and has participated, to thank you for, it's a really excellent seminar. Some great feedback coming in on the Q&A there, Edward. It's good work, nice seminar. So thank you very much for sharing that with us. Appreciate that.
Thanks.
And just for, this is the last of the QAAFI Science Seminars for this year. So the next one will be on Tuesday, the 16th of February next year. So in 2021, Tuesday, the 16th of February. And we will look forward to having you all there for that. So thanks very much for your participation today.